The overall goal of this experiment is to quantify the dynamics of cortical actin by image correlation spectroscopy, following acquisition by total internal reflection structured illumination imaging, or TIRF-SIM.
This method can help answer key questions in the biophysical field, such as what role cortical actin flow has in regulating molecular interactions at the immunological synapse. The main advantage of the technique is that it combines nanometer scale resolution with live cell compatibility, allowing smaller scale processes to be observed and analysed compared to conventional fluorescence microscopy techniques. Demonstrating this technique will be George Ashdown, a PhD student from my laboratory.
For a test sample, add 5 microliters of a 100 nanometer in diameter subdiffraction fluorescent microsphere stock solution to the surface of a fresh uncoated cover slip. Then, spread the microsphere solution over the cover slip with a pipette tip and allow it to dry. Add 200 microliters of mounting medium to the test cover slip to prepare for imaging. To begin, turn on the incubator for the stage heater of the TIRF-SIM system and fill the reservoir with distilled water to fully cover the base of the heating element. Let the water equilibrate to 37 degrees Celsius. Connect the heating collar to the objective lens and set it to 37 degrees Celsius. Then, secure the TIRF 488 compatible grading block in the light path of the microscope. To ensure that the correct optical path is engaged, switch the channel mode on the